Whenever you have section, it's a half-hour offset. You should be able to catch it. If you need to email me. We can set something up. The same thing will be true next week, because I won't be here next Wednesday. I have to leave the country again. My colleague George Bentley will be here on Tuesday. I'll be back on Thursday, and I'll have office hours again Friday, 8:30 to 9:30. So this Friday, 8:30 to 9:30 office hours, and next Friday, then we'll go back to the regular Wednesday. I think that's it. So here's what we're going to do today. So we've gotten pregnant, and now we have to decide whether or not we're going to have a boy or a girl, a male or a female. And we're going to talk about all aspects of that, from chromosomal to molecular to organ differentiation. Well, not all aspects, but as many aspects as I know something about. So we're going to talk about sex determination, a review, because we already talked about that. Primary sex differentiation, secondary sex differentiation, internal and external. And finally, I'm trying to be politically correct. I don't know the correct term. Like with atypical sex differentiation, because I didn't want to say disorders or whatever, but there's some things that are atypical that often happen. So I call it that for lack of a better term. I'm not sure if that's the right term or not. And I think we'll get through all that today. Before we get started, though, I want to tell you a story. Give you kind of a big picture perspective. Because we talk about sort of molecules and second messengers, but we really want to build that up to talk about how those molecules cause cells to differentiate and function. We really want to build that up to tissues and organs and get the idea of the big picture. So I want to give you the real big picture and how sex differentiation can affect some pretty big things. So give me a second. Okay, so here's the big picture. So I don't want to be culturally insensitive, but British food don't taste so good. So a long time, that's just how brother feels. So a long time ago, these people in Britain, but still, they needed to get like over to here, to India, where like the spices were, right? But then the Islamic Empire had all this under lockdown. So they couldn't just go like here through the Mediterranean to get to India. So you might know about Christopher Columbus and all that stuff. Christopher Columbus, funded by northern Spain, tried to get there this way and bumped into North America. And finally the solution to get to India, to get the spices, going into the 17 and 1800s, was to take ships from England, and this has a lot to do with endocrinology, you'll see, and take these ships from England and go all the way around Africa and then up to India. It turns out, though, that was a long trip. So they couldn't make it all the way. So the British took some people from some Dutch people and left them there in what became South Africa and some cows, and they set up kind of like a Crystal Burgers for what would be South Africa. Okay? But then, in the early 1800s, this guy named Shaka Zulu, because there actually technically were no black people in South Africa. When apartheid South Africa was first established, when Europeans first moved there were mostly people from a group called Khoisan. But this guy named Shaka Zulu, somewhere here north of South Africa, actually raised this huge army, which became the Zulu Nation, and forced black tribes down into South Africa. So at least 9 tribes moved into South Africa. And at the same time, the Dutch people, who became known as the Afrikaans, or sometimes the Boer, but I guess that's an offensive term, means farmer, they started running away from the British people who were managing the Crystal Burgers and moving north, while Shaka Zulu was pushing these other 9 tribes down into South Africa. And so for those of you who have followed South African politics and apartheid, the African National Congress are like the 9 tribes that Shaka Zulu pushed down. And the Inkatha Freedom Party are basically the Zulu nation, that there's often conflict between those 2 black political parties. And of course the apartheid were represented by the Dutch that were moving north. Okay, let me tell you something about Shaka. So there's one man moved millions of people and built, not just South Africa, but Swaziland, Lesotho, Zimbabwe was sort of fought for by the Indibili, which was shoot off of the Zulus. And there's a book called The Lion of South Africa. This is what Alistair Parks had to say about Shaka Zulu. Millions of people fled before Shaka's plundering regiments. Splintered groups scrambled out of the coastal plain over the jagged jaws of the Drakensberg, the mountains of dragons, running down the eastern escarpment onto the high veld. So the point is, this guy moved people all the way from as high as Tanzania. And it was his endocrinology and development that we're going to talk about today, that I would argue that's one of the driving forces that led him and shaped one of the largest and most important countries in Africa. You trust me, you'll believe me by the end. All right, we'll come back to Shaka in a second. So let me remind you as a review. In placental mammals, including us, males are heterogametic. We're XY. A Y chromosome is required for humans, for all placental mammals, to develop testes. Females are homogametic. Oh, sorry. This is one of those lectures where you're going to get a whole bunch of abbreviations that you have to learn. Sorry, this is how it is. Females are homogametic, meaning that they only have one type of oocyte. So the male, at conception, effectively determines whether or not the offspring will be male or female. And when I say male or female, I mean, in this case, whether or not you have a testis or an ovary. Kind of slow down before you guys tell me to slow down. It's a Y-dominant system, meaning that sex is not determined by X dosage. So no matter, you are not female because you have two X's. You have ovaries because you don't have a Y. In other words, if you were XO, you would be a female, and if you were XXXXY, you would be male. So chromosomally, female is the default. In the absence of a Y chromosome, you develop ovaries. That's what I mean when I say female, for the rest of this lecture. On the molecular level, it's kind of a different story. So let's start with the female situation. On the autosomes, you guys know what autosomes are? Autosomes are everything but the sex chromosomes. You have two sex chromosomes and 44 autosomes. If you're a female, you have two X chromosomes. One of those chromosomes, I'm sure as you know, at least in placental mammals, one of those X chromosomes is a bar body. Most of the genes are not expressed. Not all. There are some so-called imprinted genes that are expressed. But most of the genes on one of your X chromosomes are not expressed. The chromosome's condensed, and you don't transcribe genes from it. But it's not the same X chromosome in every cell. Yeah?
Can you repeat what you call it?
Bar body, B-A-R-R. I think you have to name that for somebody named Barr. So it's one of the X chromosomes in placental mammals, including humans, but we're placental mammals. One of the X chromosomes is highly condensed into a bar body. So for most genes, wait, what's your name?
Sharon.
Did you move?
One second.
See? I'm a spatial person. One seat and I lost it. You should see my office. It looked like a big mess, but I know where everything is in there. So in other words, for genes on the X chromosome, you don't have twice as many expressed as I do, because one of your X chromosomes is shut off in my cell. On the autosomes, well, before we even get there, we're gonna differentiate the gonads first. The gonads initially are mesoderm derived. So remember I drew this thing for you? So remember I drew this cross section? I cut myself in half? I'll do that kind of thing for you. If I cut myself in half, I got this ectodermal layer, so all of my outside skin, and you'll learn on Tuesday, things like your nervous system will come from ectoderm as well. There's the mesoderm layer. This is the layer that makes muscle, for example. And the endoderm is all the inner lining. So the inner lining of my digestive tract, the inner lining of my reproductive tract, including the endometrium. The inner lining of all of my blood vessels, that's all endoderm. And it's all surrounded by mesoderm. The reason that my reproductive tract and my blood vessels, my digestive tract can contract because it's surrounded by this muscle, this mesoderm layer. And you have to think about this as a long, stretched out donut. Think about your body as a long, stretched out donut. The things that are inside your digestive tract, in your mouth and your stomach, are outside your body. They're in the hole of the long, stretched out donut that you are. So there's a mass of mesoderm-derived tissue that buds off, that becomes the kidney, the adrenal cortex, and from that buds off the gonad, both male and female. So there's a mass of mesoderm-derived tissue that will form your kidney, your adrenal cortex. The adrenal medulla is actually neural tissue that's going to migrate into there. George Bentley's going to talk about that. But then off from that kidney adrenal complex, a couple balls of tissue bud off to form the structure that will become the gonad. And two genes are important for that. One of them you already know. One of the genes important for that is SF1, steroidogenic factor 1. We talked about that. Steroidogenic factor 1. And a gene called Wilms tumor gene 1. Named after somebody named Wilm, I guess. And mutated forms of Wilms tumor gene causes some weird kidney tumors to form. I think it's a viral version of WIT1. But if you knock those two genes out, you have no gonads. So because of these two genes, well, let's make it bigger than that. Inside your body, your abdomen, both males and females, and remember, placental mammals are the only ones that have external testicles, or external testes, which are called testicles. For all other animals, they're inside the body, and for some mammals, like cetaceans, whales. You guys don't care about that. Okay, I told you. Whales' testes are inside their body. So you develop gonads, whether or not you're male or female, from this massive mesoderm-derived tissue inside your body. Whether or not they become testes or ovary depends on the set of genes I'm going to tell you about. In the female, on the autosome, so males and females have these genes. There's a gene called SOX9. SOX9 stands for SRY like HMG box. And I'll tell you what SRY is later. HMG stands for high mobility group. And I think it just describes how the protein migrates on the gel. High mobility group, and that's the functional group that interacts with DNA. This is a transcription factor, SOX9. SOX9 is on an autosome, and it's the 9th SOX. So there's a SOX 1, 2, 3, 4, 5, 6, 7, 8. The 9th one they discovered. So SOX9 turns on a gene that you already know, SF1. SOX9 is a transcription factor that turns on SF1. Also on the autosomes. We haven't touched the sex chromosomes yet. SOX9 and SF1 together turn on a whole bunch of genes that are important for male development. Those include AMH. I'll tell you what that stands for later. Just hold it for now. It turns on P450 side chain cleavage, which you already know, right? You know that, right? It's an enzyme that cleaves cholesterol to pregnenolone. Turns on STAR, another gene that you know. And these are the genes that it takes, well, these and others, we don't know all of them. These are the genes it takes to make a testis. So by default, even though chromosomally you default sex as a female, on the molecular level, your autosomes want to make everybody male. Except that females on the X chromosome have a gene called, am I going too fast? Okay, you guys gotta say something. I'm not going too fast for me, I'm good. We're gonna do it again anyway. On the X chromosome, there's a gene called DAX1, and DAX1 shuts off SOX9. So your autosomes want to make you a testis, but your X chromosome shuts down your tendency to become male, to have a testis. When I say male and female, I mean testis or ovaries, for the point of this particular conversation. Now DAX1, you're going to love this one. DAX1 stands for this. Stands for DSS-HAHX1. Here's why. Because the gene was discovered twice independently and had two names, and so they combined it and called it DAX1. So somebody discovered a gene called DSS. A gene called DSS, dosage sensitive sex reversal, because if you're a male, if you're an XY individual and you have two tandem copies of DAX, you develop as a female even though you're XY. Dosage sensitive sex reversal, because if you're a male and you have two tandem copies of DAX1, if you're an XY individual and you have two tandem copies of DAX1, you develop ovaries. Dosage sensitive sex reversal. If you have two tandem copies of DAX1 and you're XY, you will develop ovaries. Yeah?
I guess I'm just confused why you have two copies just in X.
You have two tandem copies, so you get an incomplete crossover and you have two copies on your X chromosome. That's an XY individual. The AHX stands for adrenal hypoplasic X-linked gene 1. Because if you're missing this gene, DAX1, your adrenal glands don't develop. So this gene is also important in adrenal gland development. So two different groups discovered. One discovered a gene that if you are missing it, your adrenal glands don't develop. And another group discovered a gene that if you have two copies of it, XY individuals develop ovaries, and it turns out it's the same gene. So now it's called DAX1. Yeah, what's up?
Do females, since they have XX, do they automatically have two tandem copies of?
Two tandem means you have two on one chromosome.
So,
Yeah. So if you're XX, you got one bar body, you're only expressing one copy. Yeah.
Could you explain
Person
again?
sitting in the chair next to where Sharon used to sit.
Could you explain the chromosomal differentiation one more time for female? I understand now why molecularly males would be preferred.
Oh, not preferred. It's what I'm saying is, you are chromosomally, you are default because in other words, you're missing a Y chromosome. So the sex differentiation is by default female. But if you look at what's actually happening on the molecular level, you're a female because you suppressed your tendency to become male or to develop testes. Okay. Yes. I answered it.
Yeah.
I don't know why it's.
So for the XY, does it mean that we have, the default that we have one copy of X1?
I'll tell you exactly what's happening right now. If you're a male, and it's going to get even dumber than this and that. So if you're a male, you want to become, if you're an XY individual, you want to make testes through the same molecular mechanism, but the DAX1 on your X chromosome shuts off your SOX9, so you would develop as an ovary, except there's a gene called SRY that shuts off SOX9 and shuts off DAX1. What? No, turns on SOX9, shuts off DAX1. No, if you're a male, your SRY turns on your SOX9, shuts off your DAX1. Sorry. I drew it right, said it wrong. SRY stands for sex-determining region Y. And it was discovered because sometimes you get XX individuals that develop testes. And that's because there's a little piece of the Y chromosome sometimes crosses over to the X. And so what people did, and this gene wasn't discovered, so I was in graduate school. So people like sequenced those little regions until they finally found a region that was critical for testicular development. So sex-determining region of the Y chromosome. It's SRY, and it's related to SOX. That's why SOX is SRY like HMG box 9. Depending on who you talk to, some people will tell you that SRY and SOX, it turns on SOX9, which turns on the gene. Other studies suggest that SRY and SOX9 both regulate the same genes. So SRY is upregulating SOX9, and then they're kind of both doing the same thing. And you see, the reason that if you have two copies of DAX1, why that doesn't work, if you have two copies of tandem copies of DAX1, the SRY can't shut it all off. And you can develop as an XY with ovaries. There are two genes that are believed to turn on SRY. It doesn't automatically just come on. One is WIT1, which you already heard of. The other one is GATA4. And GATA, I think, just stands for the sequence. This is a transcription factor. It just stands for the sequence that this transcription factor binds to. So there's a promoter. Am I about to pass? So there's a promoter in the SRY that contains a GATA sequence that's regulated by GATA as well as WIT1. Now, if that's not confusing enough, and this is all a mammalian invention, right? The SRY gene is only important in placental mammals. Even like marsupials, I think they have, and I told you, monotremes, the egg-laying mammals have like 6 sex chromosomes or something. So this is only true for placental mammals. Sex is determined in other animals completely different ways. We're good? Okay, now I'm confused. Yeah. GATA4 and WIT1 turn on SRY. So it's a gene cascade. GATA4 and WIT1 regulate SRY. SRY shuts down DAX1, turns on SOX9, and together SRY and SOX9 start the cascade that made you a testis. Yeah. Or two.
Do both need to act together so that they can turn on SRY?
They both are transcription factors that regulate SRY, correct?
So if one of them didn't work correctly, then they wouldn't turn on SRY?
Usually I think if you have a requirement for a set of transcription factors, they all have to be in place. I don't know if that's always true, but that's my impression. Yes?
Is, if there's an XX individual that expresses SRY, is that sufficient to make them appear to be XY?
That's a good question. So the question is, sometimes right, the X chromosome is this big thing, the Y chromosome is this puny thing. There's what's called a pseudo-pairing region. So genes don't really cross over. I think there's only like 4 genes or something on the Y chromosome anyway. Is that right? 4? Is that what Tom Carlson told you guys? I'm gonna go with 4. So, but sometimes there is crossover. So sometimes you'll get the X chromosome has a little piece with the SRY on it. And when you get that, you can, and then when you have that gamete, you have a sperm that basically has an X chromosome with SRY. You end up with an XX individual, and 2 things can happen. One is, they usually become testis, male. But sometimes you get a mosaic because the bar body, right? So you have one, I'm not sure why it's so called a toilet paper. Maybe I shouldn't touch it. Must have been for the midterms. So, if you have an individual like that, right, sometimes the bar body can be the one with the SRY, sometimes the bar body can be the other one. So the only case of true hermaphroditism that I know of is when you have a mosaic where sometimes the SRY is shut off and sometimes it's not, and you often get a testis that's a mixture, or gonad that's a mixture of testes and ovaries.
So where does it sit?
It stays inside usually. You'll see why in a second.
And then on the flip side, if you're in an XX that has 2 copies of DAX1, could anybody tell that you weren't in, or sorry, if you're an XY with 2 copies of DAX1, could anyone tell that you weren't in XY?
If you're karyotype, I think, yeah. As far as I know, there's no other difference. All right. Well, this actually works better than the original. I feel like roll it off when it gets hurt. Okay, so there's a little bit of complication. There's a gene called DMRT1. You're gonna love this one. DMRT1 stands for and from double-sex MAP3 related transcription factor 1. It's a ubiquitous sex determining gene. Double sex was found in Drosophila and fruit flies. And double sex is alternatively spliced between male and female and can make you male or female. So double sex is a fruit fly gene. MAP3 stands for male abnormal, gene 3. And it's male abnormal because in C. elegans, Cinereptitis, however you say that, the worm, the MAP3 gene, if it's mutated, takes hermaphroditic worms and makes them turn into male. So this is a sex-determining gene that's been around since fruit flies and worms, and it's on autosomes in humans. So it's ubiquitous sex-determining gene. We're good? You understand what I'm saying? So if you're homozygous, if you're an XY individual and you're missing a copy of DMRT1, even with this, you will develop ovaries. So even with this mammalian sex-determining gene, if your DMRT1 is not functioning properly, you will become an XY with the ovaries. Yeah,
So it's autosomal?
it's autosomal. The only thing on the sex chromosome is SRY and DAX1. Everything else is autosomal. DMRT1, I'm going to do fast. Can I go now? DMRT1 works because the female is not a complete default. In females, WNT4 is important. It stands for wingless type 4. It's a gene that was identified in fruit flies. Wingless type 4. It's a cell surface binding molecule. That molecule is required for proper vascularization of the ovary. And if you overexpress that gene in XY people with testes, they'll start to develop ovary-like structures. Yeah, sure, I will. I'll put something up here. This gene WNT4 and another gene, FOXL2, stands for forkhead box. I don't know what the L2 is for. Forkhead box, because it has a DNA binding motif that's called a forkhead. WNT4 and FOXL2 are required, even with this whole system, for proper development of the ovary. So it's not completely default. You do have some genes that have to be turned on to make an ovary. Turns out DMRT1, one of the things it does is it shuts off FOXL2. And I'll try to summarize that in a minute. And then we're going to make some gonads. Sperm. Long time to make gonads. So you have this autosomal situation that's set, which actually seems like an evolutionary dead end. It's set to make everybody a male with testes. Which would be pretty hard to maintain a population that way. So you have this autosomal system that's set to make testes in everybody, but that's regulated by the X chromosome, that's shut off. You have an autosomal system in males to do the same thing. It gets shut off by DAX1, but then the SRY shuts off the DAX1. So you're a male, you have testes, because your autosomal tendency to make testes, which was shut off by your X chromosome, is in turn shut off by your Y chromosome. But on top of it, you have these genes, FOXL2 and WNT4, that even if you have the SRY system, are trying to make you have an ovary. But your DMRT1 keeps that all in check. So without DMRT1, those genes can come up and override this SRY regulated system. Yeah.
So DMRT1 inhibits both of them, not just.
As far as I know, it only inhibits FOXL2. I don't know that it inhibits WNT4. Yeah.
So is DMRT1 on the Y chromosome?
The only thing on the Y chromosome is SRY, and the only thing on the X chromosome here is DAX1. DMRT1 is on the autosome.
Then why doesn't it shut it off?
Why doesn't DMRT1? I do not know the answer to that. DMRT1, one of the known functions of DMRT1 is to shut off FOXL2, which is set to make ovaries. It may be that that's some relation to DAX or something like that. Yeah.
So why doesn't DMRT1
That's what somebody just asked. I just said if I snitch. Well, who'd you dial? I do not know. So, this is just the structure of the ovaries and the testes. What will become, as mesoderm-derived cells, what will become the Leydig cells and the fecal cells. Then, and don't ask me how, I don't know, there's a migration from something called the coelomic epithelium. So remember I told you this was the coelom? There's cells along here that delaminate. And those cells migrate into the testes. Into the gonad. I don't know how they find it. And if you're a male, these coelomic epithelial cells so to drill down into the medulla, the middle of the testis, and make the testicular tubules. They become the Sertoli cells that make up the testicular tubule. If you're a female, these coelomic epithelial cells stay on the surface or the cortex of the gonad. And they form these spherical follicles. They become the granulosa cells that make the follicles. And you see the reason that they're on the surface is because remember that egg is going to exit by popping out. If you had eggs in the middle of your ovary, they could never get out. Whereas for the sperm, you've got a whole tunnel. Remember my testicular tubules I passed around? They form like a tube. And they go all the way into the medulla, the center of the testis. Yeah?
Sorry, can you re-explain what the circles are?
But the what?
What the circles
The circles are the ovarian follicles that are formed by the future granulosa cells that migrate from your coelomic epithelium. I have no idea how they find their way into the gonads. And I have no idea how anybody knows that that's where they come from, but I'm sure, I don't know, there's some stains or characteristics that you follow to figure that out. Yeah.
Do you mind going over the male?
In the male, the cells migrate in from the coelomic epithelium and they drill all the way through the testis to make the testicular tubules. So those are the future granulosa cells. Future Sertoli cells. You still have no germ cells. Believe it or not, the germ cells come from the gut, well, it depends on the animal, the gut endoderm. So cells from inside your early gut endoderm migrate into the gonads and populate the gonads. If you're a male, they go into your testicular tubules, become the primordial spermatozoa, a primordial sperm. And if you're a female, each follicle gets one future oocyte. And the number of oocytes are fixed at birth. You don't make new oocytes. Men make new sperm every day. Unless you're smoking marijuana. I already told you about that. I don't know. But women have a set number. Females have a set number at birth. That doesn't change.
The red things were the granulosa cells, or were
The red things are the follicles that are formed by what will functionally become granulosa cells. And each follicle gets one oocyte. Yep.
Where do the germ cells come from?
They come from, in humans, I believe, it's the gut endoderm. And other animals, it comes from something called the yolk endoderm. So they're coming from inside the gut and they migrate into this gonad as it's formed. And the germ cells have no sex. They have no idea. It turns out the default is to become an oocyte. So if a germ cell misses and lands in the kidney, it becomes an oocyte. I don't know how people know that, but that's what they say. If you take a primordial germ cell and put it in a petri dish, by default, it becomes an oocyte. There's no genetic control of or whether or not you have sperm or eggs. That's controlled presumably by something from the Sertoli cells. If you don't land in the testis, you become an oocyte. So some unknown factor tells those germ cells to become sperm. Unless you're exposed to hammers. We're good? Then you have a set of internal structures